University of Florida.
Hello, everyone. I'm Gregory Schultz, Professor Emeritus at the University of Florida, where I was the director of the Institute for Wound Research. I'm going to take the next few minutes and talk to you about some exciting new developments in biofilm research. We'll begin by reviewing background data that show that bacterial biofilms impair healing and are part of the principles of biofilm-based wound care using the step-down then step-up guidelines. We'll also look at new wound blot technology. We'll look at a new chemical debriding agent. We'll look at an enzyme that breaks down the bacterial biofilm exopolymeric matrix. We'll look at a new topical point of care for nitric oxide delivery. And finally, we'll look at a novel delivery method for topical oxygen therapy. We could begin by asking, is there a common molecular pathology of chronic wounds? Now, these 4 types of chronic wounds all have different comorbidities that help to contribute to the failure of an acute wound to heal and become chronic. But what I'll show you is that there is a relatively uniform hypothesis about the chronic pathophysiology of chronic wounds. And a key part of that is that biofilms have been identified in a very high percentage of biopsies in from chronic wounds in publications, but only in a very small percent of acute wounds. And as you can see in the middle panel B, this is a scanning electron micrograph of a biopsy taken from a chronic wound. This is the surface of the wound bed, and you can see this massive structure in the middle that consists of bacteria that are encased in this self-produced exopolymeric matrix that contains unique polysaccharides, proteins, and also a significant amount of free extracellular bacterial DNA. Now, shown in panel A in this cross-section of a biopsy through a chronic wound bed, bacterial biofilms are obviously present on the surface of the wound bed, but importantly, many bacteria penetrate into the wound bed where they will also form biofilms beneath the surface of the wound bed. And as shown in panel E, one of the components that is extremely inflammatory is the free extracellular bacterial DNA shown in the red staining in panel E. And this is important because our innate immune system, our toll-like receptor 9, has developed the ability to recognize unique sequences that are found only in bacterial DNA, the so-called CpG islands. And this allows our innate immune system to rapidly respond whenever a biofilm community is beginning to form and there are components of the free extracellular bacterial DNA that become exposed. And the toll-like receptor 9 containing cells recognize that and activate and begin stimulating inflammation and at the point of the biofilm formation. It's important for us to understand also why biofilms are so highly tolerant to antibiotics. So in this example on the left-hand panel, you can see that when planktonic Pseudomonas aeruginosa are grown in suspension culture and exposed to 50 times the minimum bacterial cytoconcentration of tobramycin, within about 4 to 5 hours, all of the planktonic bacteria are killed. That's what antibiotics do. They effectively kill rapidly metabolizing and proliferating planktonic bacteria. In contrast, if we take those same sensitive planktonic bacteria and allow them to grow into a biofilm and then expose them to that same concentration of tobramycin for over 4 days, only 1 log is killed. And this shows the tremendous tolerance that the bacteria have developed to the antibiotic when they are in the biofilm phenotype. Now, a major reason why these bacteria develop the tolerance is shown in the right panel, where the Pseudomonas bacteria are transfected with a plasmid that expresses green fluorescent protein only when they're metabolically active. And what you can see is that only the metabolically active bacteria are present in the outer shell. The middle of the biofilm matrix and community is not green, therefore it's not actively metabolizing. These are what microbiologists have called the dormant persister cells. And it's important for us to appreciate that antibiotics only kill metabolically active bacteria. They kill them by interfering with essential bacterial proteins and other systems. But if they're not metabolically active, the antibiotics have a very poor ability to kill the metabolically dormant persister cells. This has led to the hypothesis of chronic wound pathophysiology, in which an acute wound experiences repeated tissue injury. Perhaps it's with pressure infusion, reischemia, and then the persistent ischemia allows for bacteria to contaminate, colonize, and convert into a critical level of both planktonic and biofilm bacteria. The presence of this bioburden stimulates pro-inflammatory cytokines such as TNF-alpha and IL-1 or IL-6, and that draws in more and more neutrophils and macrophages. They activate through the respiratory burst, provided there's sufficient oxygen available, and they secrete both proteases such as the matrix metalloproteases and elastase, as well as the reactive oxygen species such as hydrogen peroxide or hypochlorous acid. And while these proteases and reactive oxygen species do very good things when they're at the right time, right place, and right amount, when they're present for weeks at hundreds of times the concentrations that are found in non-infected healing wounds, they begin to have off-target effects, which results in the destruction of essential proteins such as growth factors, receptors, functional extracellular matrix components. And this leads to decreased cell proliferation and decreased cell migration, which we see clinically as an acute wound that's converted into a chronic non-healing wound. Thus, the key is to understand when there is a bacterial bioburden, planktonic and biofilm, and especially how to remove and kill it so that we break this amplifying cascade that results in the failure of the wounds to heal. These concepts have been brought together in the concept of step down then step up treatment that's based on biofilm-based wound care. And as you see in the left-hand column, in the first few days when a chronic wound appears, it's important to initiate multiple therapies in combination, especially aggressive debridement and use of antimicrobial therapies that will kill residual biofilm and prevent planktonic bacteria from reforming biofilm, which they can do in wounds in about 3 days. As the bioburden comes under control, inflammation drops, you can then de-escalate the treatment as the wound improves, using more maintenance debridement and also shifting to other types of antimicrobial treatments and dressings. Eventually, when the wound is healing adequately under standard of care, that's fine. But if a patient has other comorbidities that are causing the wound to not heal as rapidly as is optimal, then you can step up to advanced therapy such as growth factors, skin graft, combination cell and tissue products, negative pressure wound therapy, or as I'll show you, continuous topical oxygen therapy. And these advanced therapies will promote healing and function very well because you have prepared the wound bed correctly and have addressed the bioburden and biofilm problem. An important question for clinicians is, can you see biofilms on the surface of wound beds? And hopefully what you're going to answer is no. Most biofilms are not visible on the surface of a wound bed because, as you saw in the previous slides, the biofilm is a microscopic structure, and frankly, much of the biofilm is frequently present beneath the surface of the wound bed, where these biofilm communities are extremely inflammatory. One of the new tools to help identify where planktonic bioburden is present in and around wounds is using the bacterial fluorescence imaging. Now, this uses violet light, not ultraviolet, but just violet light, and it stimulates the fluorescence of molecules that actively metabolizing bacteria make, such as the porphyrin producers like Staphylococcus aureus or the cyan fluorescing pyoverdin, such as Pseudomonas aeruginosa. And in this study by Rosemary Hill, you can see in the photographs of the images on the right panel that in the left-hand column, you see just regular standard imaging mode, visible light, and then when the wounds are exposed to the violet light, you can see both the red and cyan, the blue-green type of staining that indicates where bioburden, especially planktonic bacteria, are present. And one thing this has helped to do is to guide immediate treatment decisions and to indicate where there are problems in the bioburden that need to be resolved. Now, the fluorescent imaging technique is very useful and has produced important clinical advantages, but it is not selective for biofilms. It mainly detects planktonic bacteria. So a new technique that has been developed is a wound blotting technique that detects only the biofilm exopolymeric matrix components. And in pivotal papers by Dr. Kojiro Nakagami at the University of Tokyo, they have developed this technique, and as shown in the upper pictures, the chronic wound bed is cleaned, and then a positively charged membrane, nitrocellulose or nylon, is pressed onto the wound bed for about 10 seconds. That tightly binds highly negatively charged components, such as negatively charged polysaccharides or bacterial DNA. And then that membrane is submerged into a cationic dye solution for about a minute. In this case, Ruthenium red, or other publications have looked at Alcian blue. Now, the key is that when these wound blots are examined, and if there's positive staining present after cleansing and debridement, then there's an important consequence. If there's no staining, then there's a 12% reduction in wound area in the week following the successful biofilm debridement. In contrast, if there is positive biofilm matrix staining after cleansing and debridement, then there's actually a impairment of healing, and the wound area actually increases. In other words, a 16% increase in wound area with incomplete biofilm debridement. Extending this even further, Dr. Wu and colleagues performed a clinical study with 31 patients to investigate the predictive value of biofilm detection for unhealed wounds at 1 month follow-up visit. What they found was that 18 cases were positively staining after the cleansing and debridement, and only 17% of those wounds were healed at 1 month follow-up. In contrast, if there was no biofilm staining on the membranes, the wounds were healing at a 77% rate at the 1 month follow-up period. Only 3 unhealed wounds produced negative staining cases, and the staining results correlated well with clinical microbiology culture assessments. In other words, there was 94% consistency, 95% sensitivity, and 60% specificity. So this indicates that biofilm infection using the Alcian blue or cationic dye staining is associated with poor healing outcome for chronic wounds. and that this is an inexpensive, rapid, point-of-care, non-instrumented test that can help clinicians decide if a biofilm is present, where it is present, and especially if it has been successfully removed during the cleansing and debridement. Now, a new agent that is available for helping to debride wounds, including biofilms, as we'll see, is a compound called Debrichem. It is anhydrous methylsulfonic acid with dimethyl sulfoxide and an amorphous silica. Now, what this does is it basically dehydrates the slough and including the planktonic and biofilm bacteria. And what Dr. Matt Malone, who's also presenting at this conference, showed was that this topical agent of the anhydrous methane sulfonic acid had incredibly effective ability to kill and remove bacterial biofilms using 2 standard laboratory biofilm models. As shown in the tables at the bottom, there was 100% eradication of the biofilms from both Pseudomonas and Staph after 30 seconds using the PEG assay, as well as using what's called the CDC biofilm reactor. So the important aspect here was that the laboratory data showed incredibly effective killing and removal of the biofilms in these laboratory models. Supporting the colony-forming units, the viable bacterial assays, you can see the scanning electron micrographs using the PEG culture well model, as well as the CDC polycarbonate disc model, showed that after treatment of either Staph or Pseudomonas biofilms, there was phenomenal elimination of the biofilm components on the surfaces of these 2 laboratory models. Importantly, this agent was also evaluated in a clinical study, and the take-home message was that addition of the Debrichem to a standard protocol for hard-to-heal wounds actually improved healing by 75% at 12 months, and importantly, it also saved over 3,000 pounds per patient because of less frequent visits to the clinic or home health care, and by more rapid healing of the wound. Another agent that is new and may help tremendously in terms of dealing with biofilms is the Dispersin B and Coacti+ technology. Dispersin B is an enzyme that was first discovered by Dr. Jeff Kaplan, and this has the ability to cleave the N-acetylglucosamine, the poly-NAG, that is a critical component of the biofilm exopolymeric matrix of many bacteria that are pathogens. This list shows the microorganisms that produce PNAG exopolysaccharide as a major component of their biofilm matrix. You can see there's lots of gram-negative pathogens, gram-positive, and also eukaryotes such as Candida. Now, interestingly, when you add the Dispersin B on to communities of biofilm-based bacteria from, for example, Staph epidermidis in the panels A and B, you can see it's the staining. But what's really interesting is when we look at this time-lapse clip, you can see that the Dispersin B just rapidly breaks down the biofilm matrix and just disperses these mature biofilm communities in the laboratory setting. Now, the effect on the number of viable bacteria of these 4 major pathogens shown with no treatment or Dispersin B, but especially when Dispersin B is combined with a typical antimicrobial dressing, that the dispersion of the biofilm matrix from these 4 pathogens leads to a tremendous log improvement in killing of the bacteria that are protected normally in the biofilm matrix. It's important to appreciate that once the biofilm matrix has been broken down, we don't want to have um, complications of impairing healing. And so in infected porcine wounds, you can see in the bottom panel with dispersin B, or just in the top panels with just a typical antimicrobial dressing, combining the dispersin B will help the help the porcine's immune system deal with the bacteria and actually accelerate healing in infected wounds. We don't want to disperse and kill biofilm, but also kill wound cells. So it's important to show that there's really no typical cytotoxicity by using the dispersin B on non-infected healing wounds. And again, you can see in the bottom panel that the dispersin B treated wounds actually improved wound healing compared to just standard care in the upper panels. Now, combining the dispersin B with an antimicrobial wound gel that was recently approved by the FDA, again, you can see that the coactive wound gel is able to effectively kill preformed biofilms even without dispersin B treatment, just by its own ability to penetrate and kill the bacteria in the biofilm. Another new opportunity that has recently been developed is the use of a treatment that produces topical nitric oxide at the point of care in the wound bed. And just as a real quick review, normally there is a significant increase in nitric oxide when there is an acute wound, starting at about a day through the inducible nitric oxide synthetase, INOS. However, when the concentration of nitric oxide and its downstream products is measured in non-healing wounds, there is a significant reduction in the amount of nitric oxide molecules, the gas, in many chronic wounds. Thus, exogenous nitric oxide gas would improve, theoretically, the healing by replacing and increasing the amount of nitric oxide available in these wounds. Now, the way this is done is just very quickly, two little foam components are mixed that chemically generates the nitric oxide. That gets trapped within these micro bubbles on the wound surface. As those bubbles break down, they release nitric oxide into the wound bed, which has multiple effects, including not only vasodilation, but as we'll see, also antimicrobial activity. And again, this is able to diffuse into the skin because nitric oxide is trapped within the bubble layer above the skin and wound bed. Now, importantly, a single treatment with the Advenox, or the nitric oxide gas generating preparation, that there will be anywhere from about a 3.5 to a 5 log reduction with 2 5-minute treatments of the Advenox material onto the surface of the mature biofilms grown on pigskin dermal explants. Importantly, the nitric oxide product does not impair healing in any way. In fact, in initial just single case studies, it's able to show incredibly promising effects on topical nitric oxide treatment. Another new product that is available to help in the war against biofilms is the topical cutaneous oxygen therapy, the Natrox device. There are multiple effects that are detrimental when there's persistent hypoxia This also includes reduced antimicrobial activity, as we talked about before, and the limited amount of oxygen diffusion into many areas of chronic wounds, and impairment in the penetration of biofilms. Shown in these images from patients that are treated with the Natrox device, either switched on or switched off, the dark red color shows where oxygen concentration is significantly increased through the delivery of oxygen through the dressing. The oxygen is being generated by a little handheld, a little belt-like cell phone device. And you can see that when the Natrox device is turned off, the oxygen concentration in the tissue rapidly drops back to non-optimal levels, but when it's turned on, you can see it rapidly beginning to increase back to levels that are important. In fact, in a randomized controlled trial of 20 hard-to-heal diabetic foot ulcers, you can see that the Natrox device had over 90% healed compared to only 30% in the control standard of care treated patients. So in summary, what I hope I've done is show you that the rapid point-of-care biofilm wound blot detects and localizes biofilms in wounds and predicts healing. The DeborChem chemical debriding agent improves healing and reduces the cost of care of chronic wounds. Dispersin B is a unique enzyme treatment to prevent biofilm formation, but importantly to disrupt mature biofilm matrix and enhance anti-biofilm treatments. The Advenox product rapidly generates nitric oxide gas at the point of care and reduces the bioburden of both planktonic and biofilm-based bacteria. And the Natrox device generates and topically delivers levels of oxygen to wounds that enhance healing by overcoming the areas of ischemia and poor oxygen concentration in tissues. This is then part of the step down then step up guidelines to